Hello, hello everyone, and welcome to the In Conversation with EBiSC, which means a project. IHI project means enabling sustainability of the European Bank for the induced pluripotent stem cell. Thank you all for being here this afternoon. My name is Angelica Valente. I'm a scientific officer at Innovative Health Initiative, IHI, and I will be moderating this discussion today. Just to give you a short introduction, IHI is a partnership between the European Union and the European health industries. And by supporting projects that brings together industries, university, SMEs, patient regulators, we aim to pioneer a new and more integrated approach to health research. Our core goals are to translate health research and innovation into tangible benefits for patients and society, and also to ensure that Europe remains at the cutting edge of interdisciplinary, sustainable, and also patient-centric health research. As most of you may know, IHI is the next generation of IMI, which is Innovative Medicine Initiative, which was a partnership between the European Commission and the pharmaceutical industries, and was focused on the development of innovative medicine and to improve the patient access to them. EBiSC2 is an IMI project and is built on the foundation laid by a first EBiSC project, which was developed during which the partners developed an initial stem cell bank containing more than 800 cell lines. But more information and details will be provided today during our conversation with the project representatives. But before we start, I would like to mention a few things. So first of all, this webinar will last 45 minutes. The last 15 minutes will be dedicated to the Q&A. We will have a Q&A session during the last 15 minutes of the event. So in case you have questions, please put down in the chat box, selecting the option panelist in order to address the question to the panelist. Also, on the event page of IHI, we have included supporting materials on EBiSC, where you can find info on the project, also the contact emails of the project and useful links. Another important thing is that the event will be recorded, and the recording will be available quite soon after the events, or maximum tomorrow morning, on our IHI website. So in case you can have it available even afterwards. So here to talk to us today about these innovative projects, we have Andreas, Julia, Stefan, and Rachel from the EBiSC2 consortium. Unfortunately, Eiko Zimmermann from Fraunhofer Institute could not join us due to unforeseen personal commitment, and he sent to all of us his apologies for not being able to attend this event anymore. And now I will give the floor to the speakers for a to quickly introduce themselves. Please, Andreas.
Yeah, hello everyone. It's a pleasure to join this I-hosted meeting. My name is Andreas Ebnet. I'm serving as a senior director in Johnson & Johnson Innovative Medicine in the area of neurodegeneration early discovery. And I had the pleasure to join EBiSC1 as well as EBiSC2 over a period of almost 10 years. So looking forward to the discussion.
Thank you. Julia?
Yes, hello everyone. It's a pleasure to be here today. My name is Julia Neubauer from Fraunhofer IBMT. I also was part of EBiSC1 and EBiSC2, and with EBiSC2 I was in a position of being the deputy coordinator and also work package leader.
Thank you, Julia. Stefan?
Good afternoon, everyone. Stefan here from Blue Oak Therapeutics, serving as Senior Director for Strategy and Enterprise Operations. In EBiSC, I actually supported the consortium representing Bayer Pharmaceuticals, so also looking forward to the panel today.
Thank you. Rachel?
Hi, everyone. Also great to be here. I'm Rachel Stieg. I also worked in EBiSC in the first project phase and the second project phase. And in the second project phase for EBiSC2 was working as the project manager for the bank.
Thank you. Thank you all for those introductions. And to start off, I would like to ask Julia, could you please set the scene explaining first what is a stem cell, just to align the audience, align everybody on this important concept, and then what is EBiSC?
Yes, of course. So I will start really a little bit from the beginning. So each of us has stem cells everywhere in our bodies. Stem cells are cells with the potential to develop into many different types of cells, and they are serving as a kind of repair system for the body. Now we are talking about induced pluripotent stem cells in our project. And these are now a more special type of stem cells that can be generated directly from fibroblasts or blood cells taken from fully consented adults. The special thing of iPS cells is now that they allow modeling of diseases, as they can be derived from individuals diagnosed with genetic diseases, for example. And later on, these cells can be converted again into almost each specialized cell of the body. That means, for example, heart cells or also nerve cells. EBiSC now is a not-for-profit central repository for exactly these kind of stem cells. That means that on the one hand, we collect lines from many different research projects internationally and worldwide, and then make them available to other researchers.
Thank you, Julia, for summarizing everything so clearly. So EBiSC developed a centralized repository for this highly useful cell. So, but prior to EBiSC, Andreas, what were the main challenges facing induced pluripotent stem cell researchers in Europe? You are on mute, Andreas.
Thanks for the question. I have to go back when EBiSC was really started, and this was back in 2014. And at that time, there were only limited access to these kind of cells, be it induced pluripotent stem cells or even embryonic stem cells. And I would like to add that there were quite some legal restriction to make use of embryonic stem cells. So for industry, for instance, there was only one way forward, and this was to go for induced pluripotent stem cells. However, at that time, it was difficult to get hands-on cell lines that had a sufficient quality control. So we were trying from the company to access academic researchers via the tech transfer offices, but it was an often frustrating period to negotiate the license. There were quite some license fees attached to it, as well as royalties. And this was ending in a laborious and lengthy purchase and access procedure. On top of it, often these cell lines that were offered by the academia or other institutions sometimes, or quite often, were lacking a decent quality control or an ethical and legal background to get access to the cells and use these cell lines. And as Julia just mentioned, this needs the consent of the donor who donated the fibroblasts, which were then used to establish the iPSC lines. So these were the initial triggers to really step into this exercise and be part of it.
Thank you, Andreas. Indeed, there are quite important, difficult challenges to deal with. But considering that, then turning to Rachel, what was the overall goal of EBiSC1 and then EBiSC2 projects?
So our overall goals really were to address all the issues and challenges that Andreas has just mentioned. So the first project ran from 2014 through to 2017. And there we were really establishing the bank from scratch. So we built an ethical and legal infrastructure to really easily be able to collect and share lines, all the processes and protocols for banking, quality control, building a website, and also an ability to distribute those lines worldwide. And then in EBiSC2, which ran from 2019 to 2023, we really looked at all of those aspects and worked to review them, improve them, to make sure that minimal updates and investment would be needed long term. So really trying to make sure that everything we built for EBiSC, the whole infrastructure, was as robust and sustainable as possible. So we really looked at everything from the ethics, the legal, the data management. We also worked on a lot of scientific development so that we could develop and share new data and also new types of cells via the catalogue. And we also looked at different protocols and different standards to make sure that what we were doing was really still as high quality as we could make it, as we could do. Both projects were a huge investment across the research community. So more than 700 people were involved in EBiSC1 and EBiSC2, and that excludes all of the depositors and all the research groups that have made lines and put them into the group. So just for building the project, more than 700 people have been involved. It's been a real investment. And I think now we are really moving into our sustainability phase.
Yes, thank you. Thank you, Rachel, for that. I would like to remind everyone that we will have a Q&A session. So in case you have at the end of the webinar, so in case you have some question, please write it down in the chat box. So now moving, because you already mentioned something, Rachel, moving on to the main goal and results of EBiSC project was the development of a sustainability induced pluripotent stem cell repository. Turning to Julia, can you please tell us more about these results more in details, and also tell us on the key results and why they are considering so important achievements?
Yes, of course, I am happy to do that. And I think at this point, we should really emphasize again our success in continuing EBiSC as a sustainable bank after the end of the funding phase. That means that we are still collecting, safeguarding, and sharing iPS lines with researchers worldwide. Up to now, we have nearly 1,000 different iPS lines in our bank, covering more than 45 genetic diseases, and these cells are provided in an open catalog. Now we do not only have iPS cells in the catalog, we are also working on providing pre-differentiated cells. For example, we already have in our catalog now the iPSC neurons, and are also working on more cell types that we would like to provide soon. And besides sharing cells, the other idea of EBiSC is to provide also data sets to the cells. That means we have genomic and clinical data sets that are available through the EBiSC Data Access Committee to a lot of cell lines that we have in the catalog. We also are offering different iPS services to the researchers, so it's also possible to directly work with us and to also include us in your proposals or in new project ideas. Another point I would like to state is our close connection to HPCREG. That's the Human Pluripotent Stem Cell Registry. That means, again, it's not only about cells, it's really about the connection of cells and the corresponding data. That is has a huge impact of EBiSC. Important to mention is, in my point of view, an ideal and perfect scenario would be in future that all iPS cells that are derived with public funding should become available to the community via EBiSC.
Thank you, Julia. And you mentioned important elements of EBiSC, the self-sustainability, but we will come back to this point afterwards during the end of our conversation. I would like now to focus more on the results. And indeed, I have another question for you and also for Stefan. Which aspect of EBiSC do you think have been most valuable and has had the most impact on research community, also industry and SMEs?
Maybe I can start at the beginning in general with the impact for academic, SME, and industry. So for us, it's really the open access to a quite huge catalog of different iPS lines covering many different diseases and genetic variants, and also gene-edited lines. All cells that we are providing are really well characterized, and we have established a special quality control to really guarantee the quality of our cells. We have also been able to streamline the ordering process and to make it as easy as possible, and to guarantee a kind of freedom to operate with all cells that we are offering. I think that's in general a huge impact to all. Now focusing more, the most impact for academia, in my point of view, is the access to support and training material. That means that it's not only about cells again. If you have questions or if you need help in starting your iPS culture, if you have questions to cryopreservation or other specific protocols, there's always the possibility to contact us. We have a lot of supporting material available. And also we give a clear guidance for onboarding of new models. So that means also if you would like to deposit your cells into the EBiSC catalog, we always support and help. For SMEs, I think the most impact is the high standard that we guarantee. So the cells that we are providing are qualified at industry standards, and also you have the ability to translate your findings to commercial applications more easily through the clear legal background of the cells. That means we have an ethical clearance, we are reviewing the donor consents and everything. So there is really a clear legal situation with the cells.
Thank you. Stefan?
Yeah, thanks. So maybe just to add from an industry perspective, so I think the greatest benefit here is the variety of the EBiSC catalog and the high quality that was already mentioned. So, but how does this benefit industry researchers, for example? Yeah. So if you think about drug development, what we often do is that we screen potential drug candidates in preclinical model systems. And in the past, those were mostly animal models that have a lot of limitations because they're not really reflecting the patient that we want to treat in the end. So now combine this with the power of iPS cells, and then really create more human or really human cell-based models. We can really learn more about the diseases we want to target and treat, and also have a higher likelihood of translating our research findings into a clinical setting, which is of crucial importance for us in industry. So that's really helpful. And on top of that, the other important piece is safety and toxicology testing. So iPS-based cells such as cardiomyocytes have become really important. And early on in the drug development process, finding those drug candidates that will cause harm to patients later and sort them out. So that's something that is really powerful if you combine iPS, or high quality iPS lines, a variety of cells such as in our EBiSC catalog with the industry setting. So yeah.
Thank you both. Actually, thank you for this quickly summaries of the results and the impact on divided them in two different categories, considering the three main stakeholders, which are very well, I have to say, based on the statistic we have, very well represented in the audience of today. We have a lot of researchers, representative of industry and SMEs. I'm sure they will be interested in these results and the impact that you have presented so far. So turning to Rachel, how will the EBiSC results be valuable and what impact will have on patient and the general public over the long term? Although something was already mentioned by Stefan, but I would like also to ask this to Rachel specifically for the patient.
Yeah, of course. And it's a really great question because, I mean, most iPSC research is aimed at better understanding of disease or developing new treatment options, especially for, diseases where there may not be currently a lot of options at the moment. So of course, the impact for patients is really important also for us. We actually did a public patient involvement session with Alzheimer Europe at the end of last year, where we worked with a group of patients diagnosed with dementia and also their carers, and we discussed EBiSC and the work that we were doing. And the overall feedback we got back was that they are hugely supportive as a group of anything that enables research tools or data to be made available that increases the access or increases the quality of research. Because of course, for patients that are donating samples to research, which is the core of iPSC research, they are donating samples because they want progress and they want new options, they want hope. So that's really important for us. In the short term, the work with Alzheimer Europe enabled us to develop an informational page for our website. So you can go on there and view it. It's called Information for General Public. It's also linked in the information package that you mentioned earlier. And that just aims to explain a little bit what EBiSC is doing in a really accessible way, also for individuals and patients that might be asked to donate original tissue samples for EBiSC. In the long term, of course, we hope that EBiSC's participation in iPSC research will make research more accessible for using really relevant human cell models, as Stefan was saying. Also that it increases the quality, the reproducibility, and that ultimately that will aid better understanding of the causes of disease, the progression of the disease. It will help find new biomarkers for disease, and it will also help find new treatment options for disease. That's, I think, all of our ultimate long-term goal.
Thank you, Rachel. Indeed, this is an important aspect for patients and for the European citizen, because if you have a very high impact on the speed and the cost effectiveness of the drug development, this is beneficial for everybody, also for the end users. So especially for the end users in terms of speed and cost for treatment that are very effective and not so expensive. So, and thank you also for talking about your project and to summarize in a really few minutes all the amazing results that you had during the project implementation. And considering indeed the results, one of the most important aspects of IMI, even now in IHI, is the outreach, and so how is the communication of your results to the relevant audience, to the main stakeholders. So considering that, Julia, how will the EBiSC2 results be shared with the academia, research center, industry, and SMEs? Although you have already mentioned a few important information on this aspect, but if you can provide more details, would be great for everybody.
Yes, of course. So due to our huge expertise in the cryopreservation of iPS cells, in the upscaling process, and also different differentiation protocols that we were able to reach within the EBiSC project, we were able to publish up to now 16 open access publications. You find them also in the supporting material if you follow the link later on. We have organized several seminars and workshops to show the EBiSC procedures, so also how to cultivate iPS cells and each necessary cultivation step of the cells. There is videos and training material available, and also a lot of protocols that are standardized, that are tested with not only one cell line, but many different iPS cell lines. So we are really in a position to say that these material and the protocols are working, and we can support also the researchers using them. Altogether, we can say, due to our 10 years of EBiSC experience, we have really a lot of lessons learned in our collaboration with many different research groups, and are now in a position to have a lot of impact and views and protocols that we are happy to share. Also to, for example, we attending a lot of different conferences. Now we also will have a booth on the ICCR this year in Hamburg. So there we always look for possibilities to get in contact and to share also our experience.
Thank you, Julia. And indeed, just to remind everybody, the supporting materials available on the IHI webpage for this event contains important links and information for people who would like indeed to have a first contact with the project, and having the possibility to search for more specific materials produced by the consortium. So going on to the next question. IMI, but even IHI, has been championing a public-private approach towards the innovative health research in Europe. And our project have consistently demonstrated that collaborative health research and also complementary skills and competences from the public and the private sectors drive important and impactful results that can benefit all European citizens. So considering that, Andreas, Julia, could you please tell us how it was the public-private partnership helping EBiSC2 project in achieving these important results that you have mentioned so far, and having the impact on all the important stakeholders for the project. Andreas, you are on mute.
I can take it from the industry perspective, indeed. I have to say that it was a very successful and collaborative spirit over this 10 years of EBiSC1 and 2, with this 2 years break in between of it. For us, it meant we could speed up the implementation, optimization of iPSC technology in our company through the collaboration with internationally renowned experts and access to state-of-the-art technology. So coming back to what Rachel was saying, the field of Alzheimer's disease, we established a wealth of cell lines that are all available now and will become available via EBiSC to the wider community, and which we are using and have used in our internal R&D activities. Another point is that we have or had access to cell lines. So for instance, if I was interested in iPSC lines or protocols to derive hair follicle cells, and you may be wondering why I'm interested in this, but this is more from a private perspective. We could just approach the EBiSC consortium and ask for support. And I'm pretty sure, Julia and Rachel, these cell lines and protocols are available. So for anyone who's joining my interest, please feel free to reach out. Then, of course, which is very important for industry, is the harmonization of the protocols and testing the reproducibility, which has been done throughout the entire process within pharma companies. And we found out that the protocols deposited in EBiSC are really reproducible across sites and across multiple applications. And finally, EBiSC, with these decades of experience in handling iPSC cells, they're just safeguarding the cell lines and the know-how. So it's easy to access this know-how now, but also in 2030, as Julia just mentioned.
So I fully agree to Andreas, especially taking into account the nice spirit that we had over the last 10 years. There was always a possibility to get feedback from the industry partners, and that is so important because the needs from private and public partners are, of course, a little bit different. And so it was so important to get this feedback and input from the private partners to really adapt our developments in terms of, for example, what kind of quality control is needed, what is really the industry standard that we are facing to, what cryopreservation formats, for example, are necessary. So it was so important to have this close collaboration, and that's really the way to ensure that EBiSC is truly a suitable resource for all iPS researchers, both commercial and academic.
Thank you both. Just a quick reminder that the chat box is open, so for questions, in case, please, we will be moving to the Q&A section of this webinar shortly. So please, if you have a burning question, now is the moment to drop it there in the chat. So turning to Rachel, so we know that the EBiSC project has opened up new collaborative and funding opportunities, but could you please tell us more about these opportunities and what is currently in the pipeline?
Yeah, of course. So as Julia mentioned earlier, one of the things we did in EBiSC2 is really to develop defined services. So we can help with more of, let's say, the routine iPSC work, the reprogramming, gene editing, differentiation, also translating protocols into different formats. And we can also help with developing more detailed things, such as, developing a differentiation protocol or new formats or methods for cryopreservation of already pre-differentiated cells. And that's kind of both sides of that have been what our collaborations have been focused on. As two examples, during EBiSC2, we collaborated with a project called R2U-Tox. So that project was working on developing pre-differentiated neurons and cardiomyocytes that are essentially pre-plated and available as ready-to-use cells. So basically making it easier for users that they don't have to do every step of the process themselves, but they can receive cells that are immediately ready for use, for example, in screening campaigns. And we've worked with that project to make those cells available as a kind of distribution factor. Another project was with the UK Dementia Research Institute, based obviously in the UK. So there they had developed or are developing a large cohort of lines associated with Alzheimer's disease with huge amounts of genetic and clinical data attached. And we worked with them to make sure that the lines could be deposited into the bank and be made available to researchers worldwide. That's ongoing, with more lines coming out this year. And since then, we've had a number of different collaborations ongoing, some with some rare disease charities who also have iPSC lines and want to make them available in the bank, and also some collaborations focused on more scientific developments, for example, developing differentiation protocols for specific cell types, looking at new methods of cryopreservation. And of course, the goal with all of those projects is that we would be able to make the outputs available via the bank, that we would be able to share the cells and the data to be able to make it available to all. So keep an eye, and we will have more updates coming this year for sure.
Thank you, Rachel. And this is the last question before moving to the Q&A section and reply to the question from the audience. The project funding is over, and we know that EBiSC2 ended in 2023. So a question for Julia: how will be the sustainability of the project be ensured over the long term?
Yeah, I really have to say at this point that EBiSC is and was a lighthouse project for Fraunhofer IBMT. So it was really for us the entry point also into the IPS community, and to get to know all these perfect collaboration partners. And up to now, there are 3 groups still working for EBiSC on maintaining the catalog and to make all the operation of the banking. That means Fraunhofer IBMT is still operating the central facility and the Mirror Bank. And as it is so an important project for us, IBMT has guaranteed to maintain all the EBiSC operations until 2030. Fraunhofer UK is operating as legal entity and managing all engagements with collaborators, users, and external partners. And the business plan that was developed within the project is, of course, very nice and is working quite nicely. And it's at the moment being implemented. And of course, we are monitoring all aspects and how it's going on to make adjustments and business decisions as is needed.
Thank you. So thank you all for presenting the project really in a nutshell, but with a lot of important information. Now we can open the Q&A session. I'm already reading some questions that we received from the audience. So first question: how do you found the bank to keep the sample free for the researchers?
So maybe I can step in here and
yeah, please
feel free to add on. So as part of our sustainability planning, during EBiSC2, we really looked in detail at all of the costs of running EBiSC. So that's the cost for staff to manage the banking, the quality control, the cryopreservation, the distribution. So we looked at all of our running costs and set a transfer value for all the vials. So it's not free for researchers, but we try and make the transfer fee as low as possible. It's a not-for-profit, and we will review as needed. The other part of the work that we did was really to try and make as I mentioned earlier, all our processes as sustainable as possible. So we really try and bring down the cost of banking, quality control as needed, with the ultimate goal of trying to keep that fee for a vial as low as possible and as accessible as possible for everybody.
Thank you, Rachel. The other question: did you reject human-induced pluripotent stem cell lines because their consent was not fit for purpose? Did you ever go back to the specific patients for reconsenting? What are your experiences regarding this? I don't know, would you like to take the floor?
Again, I will take that. That's kind of my area. So we had actually, when I look back at all the lines that we have reviewed consent for over the years, it is a really tiny minority where we have had to reject consent because it was inappropriate. I don't remember ever going back to a patient to reconsent, but I may also just have not remembered. But the majority of the time, the consent has been okay. I won't go into detail just for time. We have a publication actually that you can see listed on the additional resources that looks into some of these aspects. But we try and be flexible with the consent, whilst also trying to obviously respect the donor's consent and what they consented to make sure we still meet that threshold of fully informed consent. But we take into consideration things like when the sample was originally collected. For example, if it's a very old sample that's come from a biobank that was pre-2007 when IPS were first published, of course you can't expect that consent would explicitly state that iPSCs are going to be generated. And it's those kind of aspects that we would take into consideration.
May I add to this, Rachel? Because one of the most important work packages in EBiSC1, especially EBiSC1, was to set the legal and ethical guidelines for depositing iPSC lines. So anyone who is interested in depositing lines can approach EBiSC, because there's a wealth of information and guidance harmonized across Europe, and if I'm not entirely mistaken, also across part of the world, how to meet the legal needs to deposit cell lines.
Thank you. Thank you very much for this explanation. So we move to the next question. Do you support with developing 3D culture protocols using induced pluripotent stem cells to create organoids?
So maybe I can answer. We are using a lot of different protocols for using 3D models. So we have suspension bioreactors also in place. That means that, of course, we are absolutely supporting the idea of using organoids and producing organoids out of iPS cells from different cell types. So this is one topic that we are for the future of EBiSC. We are also looking into it because we always have to go into new directions and to not only to provide the iPS cells, but also to use them for disease modeling, for example. And here organoids are playing an essential role. And as I mentioned, we have different methods and protocols in place where we would like to collaborate also if you have a nice disease model in mind.
Thank you. Thank you, Julia. Another question is regarding the available data of induced pluripotent stem cell in the cell bank. They wrote, following the latest report from iPSCs, they advocate for an extensive proper characterization of induced pluripotent stem cells used in research. Could you please elaborate on how EBiSC approached this need for characterization data and of the available induced pluripotent stem cells in the bank, and which data is available?
Julia, do you want to take this one, or you can?
I would say just start, Rachel, and I can step in where necessary.
Okay. So the quality control has been a really big area from us right from the start of EBiSC1. So we actually did have a dedicated work package in EBiSC1, purely looking at developing a quality control regime that was robust, that picked up everything that needed to be picked up, but that was very practical, pragmatic, and doable. We developed that further in EBiSC2, and of course are working in accordance with the iPSCs guidelines. We share all the data that we can share publicly. So for example, marker expression, we share openly on the catalogue on the individual cell line pages. Any data that we would class as personal data, so for example, the SDR profiles, the genetic data, we share via our data access committee. And if you want any more information on that, just feel free to get in touch. And then we also share batch quality control data on a batch specific certificate of analysis, which is also available via our website. So I guess the basic answer is we really try and characterize each cell line batch as robustly as possible, also balancing that with the need to be sustainable. we can't possibly do every single assay, but we really try for a robust characterization to make sure the cell line has a self-renewing identity, would be classed as pluripotent, and is genetically stable.
Thank you, Rachel. I don't know, Julia, would you like to complement Rachel's answer?
Oh, I, of course, fully agreed, Rachel. The only thing is maybe to add, and we are still optimizing and working on that, especially if you're talking about the iPSC-derived cells, for example, for the neurons. We are also developing new QC panels in close collaboration with industry partners. And it's really a work in progress. So especially for the derived cell types, it's always some adjustment, and we try to be as close as possible at a kind of industry standard.
Thank you. And considering EBiSC and the European stem cell, I was expecting this kind of question. Can you comment on the contribution of your work and the assets generated in driving the 3 Rs approaches in research and innovation?
I mean, I probably can take this one because this is a question, and how is this now being used in R&D and industry, but probably also in SME, and at least for the areas I'm involved in, the iPSC lines, and we are buying and purchasing them still from EBiSC. You cannot work without them anymore. They are an integrated part of our research. We are replacing tissue culture cells more and more with iPSC-derived neurons, astrocytes, oligodendrocytes, you name it, which are all of relevance for CNS diseases. But we are using them as well, as Stefan alluded to, for toxicology assessments using cardiomyocytes. So I think, and I hope this answers the question, modern, up-to-date research in industry without iPSC-derived cell types is not possible anymore. And EBiSC was really helpful to pave the way and to establish this. And I have to thank all partners that may be listening to this webinar for their contribution, and also thank IHI and IMI for the support. And I've seen a comment from Elisabetta Vaudano, who was involved from the beginning. Thanks a lot, Elisabeth.
Thank you, Andreas. So moving to the next question. Before, Julia have mentioned the modeling disease in vitro, and they are asking to share ideas about what are currently the missing bricks in this area and the needs for the cell types and the in vitro models.
So, of course, I mean, we have now a huge possibility with EBiSC. As I already mentioned, we have more than or around 1,000 cell types with many different disease backgrounds. And of course, it's not possible for us to really have a look or use all of these cell lines, and we have to focus. And that's what we are at the moment are doing, that we are focused mainly on different neuronal cell types and cardiomyocytes to really define disease models based on the iPS lines that we have in our bank. But of course, we are always happy if consortium or collaborators are coming to us and saying we have a specific interest in exactly this kind of mutation, this kind of variant, then we are always happy to really establish and support also the protocols and to provide cells from these different areas.
Thank you. And in case, just so, because we are going towards the end of this this event, in case the audience has some other question, please, I would invite you to contact directly the consortium. Just one last question: which advice or recommendation would you give to researchers interested in using the human pluripotent stem cells in the research activities? Just quickly to reply to this question before wrap up. Would like to reply?
If I was to purchase a cell line, an iPSC line, it is looking carefully at the legal requirements, the donor consent of the lines, that you can use a cell line for your kind of R&D that you are pitching for, and high quality controlled cell lines. That's the most important one. And if you are then in the lucky position to get established and robust protocols along with the cell lines, I guess this is the way to go.
Thank you so much to everyone for joining us today. To wrap up the session, just I would like to put one final question to the speakers. If you were asked to describe this project in one sentence, what you would say, Andreas?
I think this is the most successful in terms of sustainability project I've ever been involved in. I joined a couple of IMI projects, and Elisabetta Baudano can probably say a word to this as well. So I think this is one of my take-home messages for this exercise, EBiSC.
Thank you. Julia?
From my more academic view, I have to say the development of novel and innovative iPS cells, tools, and also data that are openly shared to all is a huge success and is really the most important thing.
Thank you. Stefan?
Yeah, just happy to do so. It was huge and very collaborative endeavor with really committed people and really ensuring that iPS technology contributes to better medicines.
Thank you. Rachel?
Having a really truly collaborative and sustainable iPSC biobank, which will actually support any iPSC researcher worldwide, is really it for me.
Thank you. So thank you once again to our speakers today, our audience, for participating in this conversation with the EBiSC2 event today. If you would like to have more information on EBiSC, please, you can visit their website and also take a look at the information, the supporting materials that are available on our IHI web page. And to keep up to date with all of our IHI activities, you can visit our website, and you can subscribe to our newsletter or follow us on LinkedIn, Twitter, or Mastodon to be the first to hear about our news. And we can now close the session. Thank you again to everybody. Have a nice continuation of your day, and hope to see you soon to our next event. Bye.
Thank you all. Bye.
Thank you.
Thank you.
Thank you.